Kelly and David talked to you about data formatting, pre-processing, some quality control and normalization. So hopefully after all of that work, you're ready to actually analyze your data. And the steps of analysis that I'm going to cover today are protein roll-up. What do we mean by sort of standard experimental designs and statistics? And what do we mean by non-standard experimental designs and statistics? So we sort of left off after all of this filtering and normalization had been done. And at the very beginning, we told you that we prefer to start at the peptide level and then roll up to the protein. So there are many methods that it can be used to go from peptides to proteins. And I'm not going to go through, and specifically discuss each of these in detail. I will say that we tend to defer to the R roll-up, which is the second bullet point here. But at the most basic conceptual level, what you can think of is if I have 10 peptides that all map to the same protein, I would hope that those are somewhat close in their behavior across the samples. And so taking something like a median across all peptides to get back to the protein value might be something reasonable to do. So that's just a very basic step you can take. We mentioned earlier that you may not want to directly kind of do this if you have redundant or degenerate peptides in your data set. And the other issue that you may face is you may have proteoforms, or you might hear them referred to as isoforms. These are sort of just variations in proteins, so that it can be due to post-translational modifications, splice variants. And in order to identify these, there are some software programs that can account for these when you're like some Max Quant or other software like that can explicitly kind of search for these, but in order to do that, you have to explicitly add them to your search space or your FASTA file. And that's usually not reasonable to assume that we know what those are ahead of time. So rather than do that, what we know is that these proteins can sort of manifest in different patterns across peptides. And so we can look at those patterns in those peptides to try to map which protein or proteoform they're coming from. One algorithm that exists to do this is called PQPQ by Jenny Forshed. The reference is here. So this algorithm was developed and they look at correlations between peptides. What I'll say here is this algorithm was developed on targeted proteomics data sets, which we haven't spent any time really talking about. And the key there is that targeted proteomics data sets tend to have very little to no missing data. And for things like shotgun proteomics, like we've been talking about, where the bottom up, we definitely have missing values. So as an alternative to PQPQ, we have an algorithm called BP Quant, which is applied in PMARD-R. And the way that that algorithm works, well, so first, I guess, let's talk about why do we even bother looking for these proteoforms. So here I have an example data set where we had 50 control subjects, 25 subjects who were impaired glucose tolerant, and 25 subjects with type 2 diabetes. And we have proteomics data where we have about 12,000 peptides mapping to around 900 proteins. So that's about 2,500 peptides per protein. And what you're seeing on the right is we could make statistical comparisons of the different groups. So the IGT group versus the control, type 2 diabetics versus the control, and zeros would indicate there's no sort of statistical difference between the mean expression for that for a peptide. Negative ones would mean it would be down expressed or in the numerator, or you could think of it as up expressed. So for the second line with signature 11, there the peptide would be up expressed in the control group compared to the type 2 diabetic group. And a positive 1 means the numerator is up expressed, denominator is down expressed in terms of mean expression level. So if we looked across all the peptides, so for this particular protein, you could see there are roughly, so the signature number on the left, if you add all of those up, that's how many peptides actually map to that protein. So roughly about 260 peptides that map to the one protein. And 238 of those are just straight zeros across, meaning you don't see any sort of differential expression in those peptides. But there are 11 peptides where you see that the control group is up expressed in those peptides compared to the type 2 diabetic group. And you can see the other patterns and their frequency below as well. So if you naively sort of roll up at the protein level, or to the protein level using all 260 peptides, on the left is what you see. So 265 peptides on the left of the naive box plot is what you see for the average fold change for two of the comparisons. We would find a p-value of 0.793, so it looks like nothing is significant there. And you might say, well, some of these peptides, maybe they're like really variable, so that's why I'm not finding any significance when I roll up to the protein level. If I do that and I sort of cut the number of peptides in half based on a CV filter, and I get rid of the most variable peptides, even then my p-value comparing these different groups is like 0.931. So no evidence of anything significant. And then on the right with the two sets of red bars, what we see is that if you look at protein 1, which would be the 238 peptides that sort of match the nothing differentially expressed signature, right, your p-value is still 0.946. If I look at those 11 peptides that had a different pattern than all the other peptides that map to this protein, you can see that when you actually test for differences in groups, you get a p-value of 0.004. And what this is indicative of is that you actually have a second proteoform for this protein. And if you ignore the fact that you have that protein, you would never find that you have something that's actually differentially expressed just using a naive roll-up or even trying to account for the most variable peptides. So we bother just because we know that these things can actually happen, and that if you don't account for them, then your signal can get muted out by the other peptides that might go to a different proteoform. So you may never detect something of interest. So BP Quant is the algorithm that I mentioned. It has some key requirements. So first of all, it requires that you have at least 3 statistical comparisons of interest. So meaning you have 3 hypothesis tests that you're going after at least. And even though we have data with missing values, we need to make sure we have enough replicates that you can do all 3 quantitative comparisons. So if you have a peptide or peptides that are really sparse, they're not going to be able to conduct a statistical test for each of your comparisons, perhaps. Those are not going to be able to be used by this algorithm. So if you have a very, very sparse data set, this might not be the right road to go down. The way this works is just very similar to the previous slide that I showed you. So if we go back to this example of the different signatures that are being expressed across groups of peptides, we have the 238 group or peptides that show no sort of statistical difference. And we have 11 peptides that do show some differential expression between type 2 diabetes and the control group. And you can see on the far right column, what BP Quant does is it actually uses a Bayesian model and assesses a probability that the signal that we're seeing in this group of peptides is real. So you can see for that 0 profile, we get a fairly high probability that that is in fact a real proteoform, and that the other proteoform with 11 peptides mapping to it, we have a fairly high probability that that is also real. So in this case, BP Quant, what I'd say, you have 2 proteoforms, and it would use the 238 peptides to quantify to 1 protein or proteoform, and it would use the 11 others, and it would use our functions to quantify to a different protein. Same protein, just a different proteoform. And